Today in lab, we're going to test the effectiveness of antiseptics and disinfectants on bacteria. You're going to be working in a group. Each one of you will be testing one bacteria species. Either you will have a broth culture of Staphylococcus aureus, which is a gram-positive, Escherichia coli, which is a gram-negative, or Pseudomonas aeruginosa, which is also gram-negative. Each one of you is going to test 4 of our antimicrobial chemical agents. The 4 that we're testing today are 10% bleach, 10% Lysol, 3% hydrogen peroxide, and our own lab disinfectant, Cytocon. Other things that you're going to need, each one of you is going to need 1 sterile swab, 1 dish of filter discs. You'll each need 1 nutrient agar plate, and each of you are going to have a beaker filled with ethanol plus 1 set of forceps. The first thing you want to do is to label your petri dish. Remember, we are going to label on the agar side, not the lid. You're going to divide it into quadrants. You're going to place the numbers 1, 2, 3, 4 in the center of the quadrant. Don't forget to put your 3 initials, today's date, and just put the first initial of your test organism. To prepare a lawn of bacteria across the entire agar surface of a nutrient agar plate, you're going to want to take a swab, a sterile swab, and go ahead and open the packaging just to expose the wooden handle. We're going to do this aseptically. So take your broth culture. You want to make sure that the bacteria are mixed evenly in the solution. You can do this with your finger, or if you have a vortex handy, go ahead and give it a little mix. You have your nutrient agar plate. It's okay to keep it agar surface up. So we're going to, remember, you take your broth in your non-dominant hand, use your pinky, remove the cap, flame the lip, remove your sterile swab, and you're going to want to insert that cotton tip into the media, and you really want to let it get saturated, so leave it there a couple seconds. Flame, cap, to make sure that the whole surface gets inoculated, I like to make an X across the center. And then you're going to swab the entire surface. So you want to overlap your lines and go all the way down, and then go ahead and rotate. We're going to go all the way across. It gets a little hard when you get down, so go ahead and rotate again. The idea is you are trying to inoculate the entire agar surface. You can kind of rotate that swab as you're streaking. When you're finished, insert the swab back into the wrapper, and then this goes into the biohazard. Now that you've inoculated your nutrient agar plate, it's time to add our test chemicals. So get your beaker with your forceps, line up your 4 test chemicals. Remember that bleach is number 1, the Lysol is going to be number 2, the hydrogen peroxide is going to be number 3, and the Cytocon is going to go in that quadrant 4. How you're going to add the cleaners, you're going to add a drop to each of those discs and place the disc in the center of the quadrant. So take your forceps, they're in ethanol. You're going to sterilize the forceps by waving them through the flame. Make sure that the alcohol burns off completely before you go to aseptically take a filter disc. You want to grab it by the edge. So just grab 1 filter disc, and then you are going to add 1 drop of your test chemical. So 1 drop onto the filter disc. Go ahead and just give it a second to saturate into the filter disc. And then you are going to place that disc on top of the number 1. So this was bleach, this was number 1. So you gently place it on the surface of your agar and use the tips of the forceps to push it in place. You wouldn't take your whole lid off. I'm trying to make a video by myself. So you would use your, remember we always use the lid of the petri dish as a shield. You would place your filter disc on the agar surface, gently touch it so it sticks. Then you're going to flame your forceps again by passing it through the flame. Wait for it to burn completely off. And then you take your next disc, take 1 drop of Lysol, saturate the disc, place that on top of the number 2, or gently place it on the agar surface, touch it with the tip. And you do the same thing for test chemical 3, which is a hydrogen peroxide, test chemical 4, which is the cytokine. If you want to take a break in between the chemicals, you can just put your forceps back into the beaker of alcohol.